am Dr. V. SUMATHI, Assistant Professor, Department of Microbiology and DD Coordinator in the Annamali University of Department of Microbiology. My topic is bacterial internal cell structure. Let us see the bacterial internal cell structure. First, what is the structure of bacteria? The bacterial structure is a very simple structure. When compared to eukaryote, it is a very simple, and this internal structure does not have any internal organelles like a eukaryotic cell. All the activities are mostly that is performed in the internal cell of the bacteria. So, here there is no any that is most important organelles in the bacterial cell except the chromosome. So, all the activities mostly takes place in the special structure of the cytoplasm of the bacteria, and this bacteria, it is a small size, simple design, and it carry out the broad metabolic capabilities of the bacteria to carry out the simple growth, and easily it can divide in the simple environment. So, it requires only simple nutrients from the environment. They were first seen under the microscope, that is the bacterial structure it can seen under the microscope. Their microscope was developed by the Antony von Leeuwenhoek in the year 1676. The most microscopes are very useful to the we easily understand or easily we can see the structure of the bacteria. This is the structure of bacterial cell. It having the ribosome, inclusion bodies, flagellum. So, it having the internal external structures, and also some bacteria having the cell envelope. This is the structure of eukaryotic cell. So, mostly the bacterial cell structure that is organized into three categories. One is the internal structure, another one is the cell envelope, and one more thing is the external structure. Internal structure, that is cytoplasm, nucleus, chromosomes, plasmid, ribosomes and storage granules. Cell envelope, that is covering layer, outer covering layer is called the cell envelope. It is cell membrane, peptidoglycan cell wall or outer lipid membrane. Mostly lipid membrane, that is mostly found in the gram negative bacterial cell. External structure, that is present in the outside of the cell, flagella, fimbriae, expili and glycocalyx. First, internal structure. Now you see the internal structure. It includes the cytoplasm, chromosome, plasmid, ribosome and inclusion bodies. First, cytoplasm. What is cytoplasm? Cytoplasm, that is present inside of the bacteria. So, it is a broad space in the center of the bacteria. The portion that is a cell, it lies within the plasma membrane. So, inside of the plasma membrane, it is called a cytoplasm. So, excluding the genetic material, the plasma membrane substance, it is called a cytoplasm. Mostly the cytoplasm is a gel like matrix. It is composed mostly water, enzymes, nutrients, waste and gases. And what are the cell structures? So, inside of the cell present in the cytoplasm is the ribosome, chromosome, plasmids, as well as the mostly the important that is metabolic activities takes place in the center of the cytoplasm. We can see the some granules under the electron microscope. These mostly granules are very small size. It relatively featureless, that is it does not having any important functions. It carry out very important functions, that is the cytoplasm carry out the growth, metabolism and replication. Another constituents, that is the proteins including enzyme, vitamins, ions, nucleic acids and amino acids and their precursors, sugars, carbohydrates, fatty acids and their derivatives. So, these are the constituents of the cytoplasm. Nucleus. So, nucleus, that is in bacterial chromosome is called a bacterial chromosome or bacterial nucleus. In the eukaryotic, it is called a nucleus. So, eukaryotic cell is having a outer membrane, but this bacterial nucleus does not having any outer membrane or outer cell wall. The nuclear region of cytoplasm where the chromosomal, it is called chromosomal DNA or plasmid DNA. It is mostly located in the center of the bacterial cytoplasm. It is not a membrane bound nucleus like in eukaryotic. The bacterial cytoplasm membrane does not having any nuclear membrane, that is it is called a nucleoid or DNA genetic material. plasmid. The another one is the plasmid. This plasmid is present in the cytoplasm of the bacteria. Plasmid is a extra chromosomal DNA and it is a closed circular DNA. These mostly plasmids involved in the antibiotic resistance or virulence activity. Structure, that is, this plasmid structure is similar to the bacterial chromosome, but considerably it is smaller when compared to chromosome, it is smaller. In a few species linear plasmids have been found. So, sometimes it is in circular form in the some organisms, in some organisms it is present in a linear form. The size of the chromosomal DNA is typically 4000 kilobase. Plasmid DNA ranges, that is it is varies among the organisms 1 to 200 kilobase. Number of plasmid, that is varies, the copies varies from 1 to 700 copies of plasmid in a cell. Plasmid function. What is a plasmid function? Already I told it is mostly involved in the resistant activities. The plasmid mostly the function is not always known, but they are not normally essential for survival of host. So, without plasmid the bacteria can survive and they are mostly involved in the function, that is antibiotic resistance. Antibiotic resistance is a big advantage for the pathogens. It protects the its pathogen from the host. Host means animal, human or any cells. Mostly it involved in the virulence character. Virulence character means it having a capacity to cause a disease in the host cell. Example, E. coli plasmid, entero, that is a P307, it codes for an enterotoxin. Enterotoxin, it is produced by the E. coli, it is a pathogenic and cause a enteric infection in the human beings. Conjugative plasmid, this allows the exchange of DNA between the plasmids, bacterial cells. Conjugative plasmids is a sex plasmid, so it transmits the genetic material from one cell to another cell. Mostly the plasmids, that is transferred from one bacteria to another bacteria, so the plus cells to and minus cells. Donor to recipient. Plasmids are not involved in reproduction. So, it is a it is atomically replicable, that is self replicable. It does not depend the chromosome for their replication. The plasmids are passed to other bacteria by two means. So, one organism to another organism by the way conjugation tube way. For most plasmid types, copies in the cytoplasm are passed on to the daughter cell to, that is especially from the mother cell to daughter cell. So, this transmission, that is during the binary fission. Binary fission means that is each cell that is divided into two cells, that is called a binary fission. The other type of plasmid, that is like pilus, also present in the bacterial cell. So, it called sex pili. So, sex pili means it involved in the during the conjugation, it transmit the genetic material from one cell to another cell. It is mostly plasmid is a instrumental genetic material. So, it transmits the one genes to another genes by the use of the conjugation tube. So, it is a important useful tool in the genetic engineering or in the recombinant DNA technology. So, gene that is developed by using this technology by the recombinant DNA technology. So, mostly it gives the resistance to the heavy metals and it gives the virulence factors against the host and cause infection in the animal or plant. Next one is the ribosome. Ribosome mostly it involved in the protein synthesis. It consists of RNA and protein. Mostly it present in the cytoplasm, that is in the abundant, often grouped in long chain called polyribosomes. It gives the cytoplasm of bacteria a granular appearance in the, that is under the electron microscope it gives a granular appearance. These ribosomes are very small when compared to eukaryotic cells and also it having a similar function, but the action, that is the function is the same as a eukaryotic cell. Bacterial ribosomes have sedimentation rate. During the certification, it gives the sedimentation rate, that is 70 years ribosomes. Especially it gives it having a two subunits, that is one is the larger subunit, another one is the smaller. Larger is the 50 years, smaller is the 30 years. The unit used to measure by the, how can it is a measure means by the sedimentation velocity. This sedimentation velocity measurement was done by the, it is that is introduced by the Svedberg. next one is that the ribosomes are mostly, it is a nucleic acids and it is a building block of proteins, amino acids and building block of proteins. The bacterial ribosomes are similar to those of eukaryotic, like a eukaryote, and it is a smaller, already I told. The next one is the bacterial ribosomes are never bound to other organelles as they sometimes, so it does not have any outer structure or any organelles. So, it is a free form, it present in the cytoplasm of the bacteria. There are sufficient difference between the bacterial ribosome and the eukaryotic ribosomes. So, mostly these ribosomes are, it inhibit the functioning of the that is the antibiotic. Mostly we are using some antibiotics, that antibiotics it stop the function of the ribosomes in the only in the prokaryote, not in the eukaryote cell. So, it disturb the bacterial cell wall, bacterial ribosomes, that is protein synthesis, it not disturb the eukaryotic host cell. Mostly example the streptomycin, it is antibiotic, it binds to the 70S ribosome. 70S means I told you that is it present in the bacteria. 80S ribosome, it is present in the eukaryotic host cell. So, it disturb the 70S ribosome of the bacterial cell, it stop the protein synthesis. Bacterial chromosome, the another one is the bacterial chromosome, it is otherwise called genophore. The bacterial chromosome consists of a single circle of deoxyribonucleic acid, that is DNA, it is a genetic material, it present in the center of the cytoplasm. DNA mostly it is a double stranded, that is double means two stranded, line up anti-parallel to each other and the bases are linked together with the hydrogen bonds. So, each DNA strand are linked by the hydrogen bonds. It includes most of the genetic material of the organism, mostly it is a genetic material of the whole organism. Bacterial chromosome is mostly in the eukaryotic cell like DNA, this one also DNA genetic material in the bacteria. So, this genetic material it is a form in the brief form without any outer structure, but the that is eukaryotic cell it is having a outer membrane. In many bacteria the DNA is present as a single, mostly it is a single form, circular form, maybe in the linear form. In some bacteria it is present as a linear form. Some bacteria having many, maybe it is 2 chromosomes also present in some bacteria. The another one is the inclusion bodies. Inclusion bodies bacteria can have within their cytoplasm. So, the inclusion bodies that is present in the cytoplasm of the bacteria, it is mostly functionless, it does not have any important functions. Some are called granules and some are called vesicle form. So, it is present as a vesicle pouch-like structure, it is called vesicles. Inclusions are considered to be a non-living compounds, it is does not in the living condition, it does not possess any important metabolic activity, but it is mostly it is present in the cytoplasm during the starvation conditions. So, at the time these inclusion bodies are utilized by the bacteria. The most common inclusion what are that is the glycogen, lipid droplets, crystals and pigments. The another one is the granules. First granule, what is granule? It is a densely compact substance without a membrane covering. It does not have any membrane covering, it present in the form of the granules. Under the microscope we can see it is in the form of granular form, it is called inclusion body, granular inclusion body. Nutrients and reserved may be stored in the cytoplasm. This one already I told. So, it is stored in the cytoplasm. When the starvation condition, these nutrients are utilized by the bacteria, lipids or polyphosphate, glycogen or these are the foods are utilized by the bacteria during the starvation condition. Nitrogen also mostly used for the growth of the bacteria during the starvation. Each granule contains specific substance, that is glycogen, polyphosphate. So, these are mostly it gives the energy for the organism. Sulfur bacteria contains reserve the granules of sulfur. So, sulfur bacteria it reserve the sulfur and store the sulfur granules in the cytoplasm of the bacteria. These granules are depleted in the starvation. Next one is the vesicles. Vesicles, these also inclusion bodies, it present in the cytoplasm of the bacteria. So, these mostly vesicles are used for the especially in the swim in the water, especially in the marine water, the most of the bacteria cytoplasm they have the vesicles to swim. So, this swimming that is move towards the light and nutrients. Example, Magnetobacterium bacterium, Aquaspirillum magnetotacticum. It stores the magnetite. So, towards the magnetite it acts, and also it utilizes the ferric oxide for their survival. Next one is the micro compartments. Micro compartments are widespread membrane bound organelle. So, these compartments are having a outer covering layer, that is called membrane. It is a membrane organelle, and this membrane organelle are made up of protein shell. It includes the various enzymes, carboxysomes, magnetosomes. Carboxysomes is a protein enclosed bacterial microcompartment. Magnetosomes also it present in the bacterial cytoplasm. It contains magnetic crystals. Carboxysomes mostly involved in the carbon fixation. During the carbon dioxide or carbon fixation, these carboxysomes are involved. Thank you.